My topic today is just an overview of medical and population genetics with a particular eye towards the genetics of complex traits that are influenced by both many genes and the environment. I have some slides and I'll go through them, but if there are places where it doesn't make sense or people have questions, please shout them out. As you may know, in this forum we typically interrupt and ask questions, and it's more important to get at the underlying understanding and that we're really reaching some agreement on that rather than to get through every slide. So in some sense, the study of human genetics and medical genetics really starts with a simple and fundamental observation, which is that diseases tend to cluster in families. And this observation that we all know at some level because people just have families and they recognize that there are sometimes diseases that aren't that common in the population, or maybe even are common, but are even more common in a given family. And this has been codified by what's called classical genetic epidemiology, where people just very carefully look at the rates of diseases in well-matched samples, for example, in the population at large, and then in one frame in the families of people who have disease and in relationship to the genetic ancestry. So how distantly related are people and how similar is their rates of disease. And there are some diseases, classic single gene disorders, for example, and I'll use cystic fibrosis here as an example, where the disease is relatively rare in the population, much less than 1% frequency, where the risk to a sibling is dramatically higher, and as we'll discuss briefly, in this case follows Mendel's ratios, where it's 25% in the sibling of someone. This is a classical recessive disease, and such that homozygotes tend not to have children. So there's an obligate heterozygosity of both parents, typically of the parent of someone with cystic fibrosis, giving a 25% frequency in the next child of having cystic fibrosis. And then in an identical twin, the likelihood of having cystic fibrosis, if one twin has it, is essentially 100%, because what's known as the penetrance, which is the idea of, given a certain genotype, what's the likelihood that the phenotype will manifest, is extremely high for this genotype. So what this tells us, beyond any reasonable doubt, is first of all, that inherited DNA sequence influences risk of this disease. And you might imagine, and I'm not going to go through it in detail, that you could say, well, how do we know from this kind of data it's not actually shared environment? And the answer is, from this alone, you don't know that. But there are actually, if you want to get at that in the most clear-cut way, there are actually extensive studies, for example, of identical twins who were raised apart, separated at birth, and of adopted children brought into the same house as someone with disease. And those studies typically show very low or non-existent clustering of disease in those, for example, adopted children brought into the same house, and similar or indistinguishable rates of clustering for identical twins raised apart, typically, as those who were raised in the same household. And so this says, again, that inherited variation in DNA sequence influences risk of this disease. And because it follows Mendel's ratios, without knowing anything else, without doing any molecular genetics or genome sequencing or anything fancy, you know that there is a single gene in this family in which a highly penetrant mutation is necessary and sufficient in this family to cause the disease, because it follows Mendel's ratios, and therefore the genotype is tracking perfectly, the phenotype, I should say, of the disease is tracking perfectly with that genotype in the population. You don't need to know the genotype to know that that must be the case. Common diseases, here arbitrarily defined as those that are present, let's say, at more than 1% frequency in the population. These common diseases make up the vast majority of hospital admissions and of healthcare costs, because as you all know, common things being common, it turns out that although rare diseases individually contribute to a substantial burden of disease, it still remains the case that the 20 most common diseases make up the vast majority of all medical encounters, morbidity, and mortality. So if you take a disease like type 2 diabetes, and I'll use it as an example in my talk simply because it's what my lab works on, and so I have the data easily at hand, but I think the patterns, although specific to each disease, are broadly comparable. You see here, first of all, the risk to the individual in the population is much, much higher, because this is a common disease. So in America today, and it depends on age group, and it depends on the population, the subpopulation of which you're a member, but say on about a risk of 10%, let's say, for an adult to have diabetes in America today. So that's, first of all, much higher. If you look at the risk to the sibling of someone who has this disease, it's 3- or 4-fold higher, say 30%. And if you look at the identical twin, it's much north of 50%. Some studies where they follow people for a very long time, it's as high as 80 or 90%. This also tells you, just as in the previous example, that variation in DNA sequence influences risk. It tells you that variation in DNA sequence does not perfectly determine the outcome, because identical twin concordance is not 100%. It tells you, without doing any fancy molecular genetics or linkage or association or genome sequencing, that there's not one gene that explains this, because it doesn't follow Mendel's ratios if you go to greater extent. And it also tells you, if you do other kinds of studies, that there is an impact of environment and behavior in this trait as well as of genotype. So all these things, and I'm going to use a historical frame in this introductory talk, all of these things were very clear a long time ago. And in fact, I'll build this argument that many of the things that our field continues to debate about the nature of the genotypes underlying complex phenotypes actually were debated sometimes with as much clarity 100 years ago as they are debated today. And so when there are questions that continue to be debated even after facts become clearer and clearer and clearer, you realize sometimes there's a philosophical underpinning rather than an empirical underpinning. That where the truth is somewhere in the middle, people polarize to the extremes simply because they like one end of the extreme versus the other, or somehow it scratches their itch, rather than simply accepting that there is a much more complex problem. It reminds me a little bit, when you watch this later, if any of you were watching in the ether, we're living at the time I'm giving this talk through the Democratic and Republican national conventions for the election of the president in 2012. And you can see, if you watch these two, there's one country we live in with one reality, and somehow listening to these two conventions, you get the idea that things are really very different. And I often feel that way in human genetics, that the sort of Mendelian world, I'm not kidding actually, that the sort of Mendelian worldview that wants to say there's a rare genotype, underlying every trait, and maybe the alternative extreme view that everything is infinitesimal alleles and there are, indescribably complex. These are two extreme views, and the reality, of course, is some of both and somewhere in the middle. So why do we care? Well, there's lots of reasons we might care, and one of the things about human genetics is that it's a bit of a Rorschach test, because some people care about human genetics because they're interested in human history and evolution, and human genetics can teach you about that. There are some people who are interested in human genetics because they're interested in understanding the underlying basis of disease and biology. There are some people who are interested in genetics because they care for the individuals with these terrible, rare genetic diseases, and they want to have better solutions for their patients. And there are some people who want to identify how to target therapy for common diseases. And all of these things are potential outputs of human genetics, and so it's good, it's all well and good that we care about these different things. But as we think about our field, it's important to realize that all of these exist, and the right answer under one of these questions might be the wrong answer in a different question. So all of these, from the perspective of medicine, require that we can somehow get from what's depicted on the left in this slide, which is a population of human beings, free living, related to one another as they may be in a free living population, living in the free environment, behaving as they choose to behave, and who have different disease outcomes, here blue and gray just to represent some disease or trait of interest and not. And we want to get, on the other hand, to improved diagnostics, prevention, and therapy. And so one of the very special things about human genetics is that it offers an approach to go from the left to the right, to start with the patient and the population, and to try and discover, without any preconceptions other than that there are genetic variants that influence the trait, what the underlying biology may be, and take the long road. And note, this is not a shortcut. This is a long road to go from the population to understanding the biology to then coming up ultimately with better diagnostics and therapeutics. And this is a many-step process, but I'm going to divide the steps into two categories. There is the first step, which could be called, and is called, genetic mapping, which is the idea of starting with some individuals in a population, and by the way, those individuals might be in families. In fact, they are in families. I guarantee you those individuals live in families, but you might choose to study them in families, or you might take one member of each family. Again, these are choices you might make. And to go from those patients to the genes that have variations in them that in the population or families you're studying contribute to variation in the trait. And you can do this without having to make any preconceptions about what the underlying nature of those genes may be in terms of their names, or the belief system of your field in terms of what the biology is. And this is extremely special. It's special because, first of all, it's unbiased with regard to the names of the genes, but there are other unbiased genomic approaches you can do, for example, in vitro, in cells in a dish or in a fruit fly. But second, it's done in the human population. And there are other things you can do in the human population, but they don't have the advantages that DNA sequence studies have of being unbiased with regard to the biological hypothesis, and also, as I'll get at, being able to establish causality as opposed to just correlation. And so these two things are very special about this approach. But in our enthusiasm to identify the genes underlying traits, rare and common, simple and complex in inheritance, sometimes it's not always discussed as much that this is simply a starting point. That in fact, if this enterprise, which has been really racing ahead over the last 30 years, when it completes its work, and I don't think that day is so far off, where we have a very comprehensive, I didn't say complete because nothing is ever complete, but a very comprehensive mapping of the diseases in the population, to DNA sequence variants and genes that influence those, and vice versa. You can go from any gene or variant to whether or not it affects a phenotype. When we have that very extensive compendium, all we have is a compendium, right? Because we don't understand what genes do for the most part. We don't have the ability to look at DNA sequence changes and to predict their effects on the molecular function of that transcript, if it's in a transcript, or that non-coding region, if it's non-coding. But even if we did know molecularly what it does do in the context of the circuitry of the cell? And even if we knew what it did in context of the circuitry or the pathways of the cell, what does it do to the physiology of the cell? And even if we understood what it did to the physiology of the cell, what does that do to the physiology of the organism? And even if we understood what it did to the physiology of the organism, what does that mean to the population ecology of that individual in the environment in which they live and the exposures to which they're exposed that ultimately leads to disease? That all I'll broadly call pathophysiology, understanding how these alterations lead to disease. And if you understood the genotypes and the pathophysiology, then I think you would be in a position, and you're going to do that in an iterative manner often. It's not that there's any royal road. It's not that one will always lead to the other. You would then be in a position to develop much better diagnostics, preventatives, and therapeutics, at least much more rationally designed diagnostics and therapeutics, because you'd understand what was going on. Now, when you discuss it in this way, some people they start to lose interest because they say, that sounds really complicated. That's never going to work. But then I always say to myself, well, what's the alternative? The alternative that people would often propose is, instead of starting in the human population and doing genetics, which, by the way, there are alternatives and they're very valuable and we should do them, but the alternatives are not necessarily simpler. They're just simpler for you as a basic scientist. Because what you might do is say, well, I'm going to start with a cell in a dish that is a model of a disease, or I'm going to study with a mouse or a fruit fly or a worm or whatever I'm going to do, and I'm going to understand that. I'm going to do the genetics and pathophysiology, whatever I do, but then you've got to figure out how to connect it to the human. And what people often think is, well, I'll publish my paper in Cell and Nature, and some other people will go develop a drug or do human biology and figure out if this is relevant. But then you get back to this very complex set of challenges of how are you ever going to do that in the human population? So sometimes if we think through the entirety of the process to diagnostics and therapeutics, and we recognize there are many other inputs to this, it's not just a purely human genetics approach, we inevitably have to understand this set of relationships of human pathophysiology and all its complexity to disease if we want to have anything other than a serendipitous approach to treating patients. So I've already discussed what it is that human genetics can contribute, and I'm going to skip over this because I've already mentioned this, but I want to make one other point that I sort of pointed to earlier, which is that there are many different designs of human genetic studies. Some of them are done by pre-selecting in the population those individuals with very extreme and unusual phenotypes that have the characteristic that they travel in families, let's say, according to Mendel's ratios, and then you study those. And that's a very good approach. That's a traditional human genetics approach, and it's a very good and solid approach. And it will lead to, in this picture of the elephant, and this famous idea that there's one elephant you're trying to understand, which in this case, the elephant I'm trying to understand is the basis of the disease. So that approach will lead you to one solid trunk of the, part of the trunk or the legs or the front or the back of the elephant. Other people like to say, I'm going to take the average patient in the population who has disease, who also shows heritability, and I'm going to do a genome-wide association study with common variants, or I'm going to sequence now today we can sequence all the exons of their genomes, or I'm going to do something else. That also has proved productive, and we'll also discover things. And that's a different part of the elephant of that same disease, if that disease comes in these different flavors of common and rare. Some people like to study candidate genes that come out of other biological approaches. All of these are different methods that are aiming to understand the pathophysiology of this disease in its different forms and due to different perturbations. A rare mutation of large effect is a different sort of perturbation that can lead to the same endpoint perhaps, or a different endpoint that shares some features as another kind of perturbation of the system caused by genetics. And I find this actually, although obvious, a very centering kind of conception as we talk over the course of this year and in our field and in our careers about human genetics, because you will find as different speakers you listen to and in your own work that often if you've spent the last 3 years studying the foot of the elephant, you tend to say this is what elephants are. And the most, or scientists actually often like to say, and I think it's a huge problem actually in science, is instead of saying I've studied the foot and here's what the foot is and there may be a rest of an elephant, I didn't study it, but I understand the foot pretty well and everyone should be able to applaud that, they'll say, I'm going to try and convince you, you should never try and convince people of things in that way, I'm going to try and convince you that the foot is the master regulator of the elephant. Without feet, you can't walk. And if you can't walk, you will not be able to eat, and if you don't eat, you die. So feet are the most important part of the elephant, and I'm going to tell you all about feet, and somehow discount the rest. All right, and that's just, I think that's counterproductive. So I think in our field, we actually suffer from that as much as any field I know, because the different people who take different approaches often focus on that and demonize the other approaches, as opposed to say they're part of a continuum. And I very much my message in this admittedly superficial overview talk is to realize that if you're studying a disease, and I'm going to focus largely on that, although there are these other questions like ancestry and evolution and lots of other important things, but if you focus on a disease, you should realize that any genetic study or non-genetic study that gives insight into the biology or the disease pathophysiology that you're interested in is relevant to what you're doing, and that your challenge should be to figure out how do you synthesize these into a coherent view rather than to say, the approach I've chosen gives the important answers and the other ones I will conveniently ignore. Okay, so I'm going to step back now with that as an introduction and give a historical overview of 150 years of human genetics. Again, admittedly very superficial, but at least where some concepts come from, so that as over the course of the next 6 months of this series and the course of the medical and population genetics meetings over the year and everything else that you choose to do, there's some historical context. So although it is the case that there is actually writing that goes back thousands of years to ancient Greece about familiality of disease, and in the Bible there's talk of if multiple sons bleed during circumcision, that you cannot circumcise in the Jewish tradition other sons because they recognized there was something like hemophilia. Although that goes back thousands of years, the codification of genetics as an idea I think can be traced to Mendel, who famously studied peas, as you know, and other plants in this abbey, which is in Brno, Czech Republic. And he grew the peas in this garden right here. I went and visited this, remarkably enough, on my 20th wedding anniversary to the day with my wife, Jill, who's a wonderful wife and mother and friend. We were in a trip to Vienna and Prague and Budapest. And I recognized that we were going to be in a train going by Mendel's Abbey on the actual day of our 20th anniversary. And I said, could we get off the train and go visit Mendel's Abbey? and she's actually said yes, which was quite nice of her, and actually it turned out to be very interesting. But anyway, this is Mendel's Abbey, where he worked, and he famously recognized the ratios, the mathematical ratios of the inheritance of wrinkled and smooth and yellow and green and other peas and other such traits. And actually in the museum, which you enter through this door right here, it points out, which is actually quite interesting, that not only was this the codification of genetics, but that it could be seen as the beginning of mathematical biology. Because I guess the idea at that time of putting mathematics and mathematical ratios into biological naturalism, you know, sort of studying the living, the world as that tradition described, was actually really a new idea. So it's quite remarkable. And the other really cool thing about this museum is they have a big display about the sequencing of the human genome. And so it was very exciting sort of to work in a place that had contributed to that, and in a field, and then to sort of see the beginnings, and then at least this way station. So Mendel's work, by the way, was lost for 35 years, cited 3 times before 1901. He wrote 35 years before that, and he was long dead before his work's importance was recognized. It's also worth noting to the young people in the audience, soon after doing this landmark work, he was promoted to being the chair of the department of Brno. Actually, no, he was actually made the head of the Abbey, and then he never really could do science again because he was too busy dealing with the administrative burdens. This is actually a true story. So there is a, who knows how much the world would have known if, that's why they promoted him, to have him stop with these ideas. But anyway, but in 1901, his work was rediscovered, and the rediscovery process is very interesting because what happened was he published work, and it's not that no one knew, actually. There were people who were aware of his work, but the question of how traits varied and what the underlying base of that was not a question in the 1860s that anyone was interested in. So the answer to the question was seen as uninteresting, or just not uninteresting, but like they were disinterested. You know, just it had nothing. It meant nothing to them. They had no interest, no opinion. Okay. But then 35 years later, really following on Darwin, because when Darwin talked about, natural selection, that was of great interest immediately to people. But if you realize, if you think about it, natural selection requires that there's variation in traits, and that the variation is passed along to offspring. If there's no variation in traits, and if there's no ability to pass that on to the offspring, then there is no natural selection. Okay? And actually, Darwin recognized this, that his idea really had 2 elements. It had the element of variation in traits that was passed on, and then natural selection on the traits. And the natural selection part was sort of obviously right, but he had no explanation of how that variation in traits worked. And I'll come back to that in a few minutes. So after many years of people thinking about Darwin, they were interested now in where trait variation came from and how it was passed along. And so when Mendel's work was rediscovered, it instantly then was a sensation, because people said, okay, this might be how that question we become interested in might be resolved. And it's worth noting that the most widely used genetic test in medicine, or one of the, if not the most, which is blood group antigens, which is a genetic test. It's not a DNA-based test, but it's a genetic test. Because blood group antigens, ABO blood type, is not determined by behavior or environment. It's determined by inheritance of a single codominant locus in the human genome with full penetrance. And this was recognized over 100 years ago, in the first decade of the 20th century, that this was a genetic trait, that it was relevant to medicine, and it's personalized the ability to do blood transfusions, right? Because if you didn't have, this is perfect personalized medicine. If you don't have, no it is, if you don't have an ABO blood type known, and you were to do a blood transfusion, most of the time you're going to harm or kill the person. And yet if you have the personalized genetic test, you will not. You will give them an absolutely safe and effective clinical intervention. But no one thinks of this as personalized medicine, and no one cites it as personalized medicine. Why do you think that is? Why do you think that is, actually? Do you think of it as personalized medicine? When you read reviews about personalized medicine, do they talk about ABO blood groups? They don't. So I think one reason is you don't measure the DNA. Okay, it's not, so sometimes when people say personalized medicine, what they really mean is code for, I like DNA sequencing, or I sell a DNA sequencing machine, or I study DNA sequencing. And therefore, reading out the DNA is sort of the idea of the digital code. Is this idea, it's an idea. You know, it's really attractive. It's gonna be like digital, and like the internet, okay? And there is an element of that, by the way. But here it's a phenotypic test. And it turns out, I think in medicine, hemochromatosis is another good example. Often, even if genetics leads you to a deep understanding of discovering the genes and how they work, it will often be the case, I think, that in clinical medicine, a phenotypic integrative readout of that pathophysiology may be a better test in some cases than the DNA. In other cases, it may be the DNA. Time will tell. But we shouldn't assume just because we use DNA sequencing or methods derived from that to do research that it necessarily is the case that we will use DNA sequencing in the clinic, or that because we can collect DNA sequence information, it will necessarily be the best way to figure out in the patient what's going on. So it could be there are things we can do phenotypically in the patient that integrate all that genetic information and environment and behavior in some way that's better off. Nonetheless, incredibly important clinical intervention, genetic at its core. But the trouble with this kind of story is that you had to actually discover the phenotype of the blood group incompatibility and then understand the antibodies and all this in order to figure it out. It wasn't a generic approach to go from a phenotype to its underlying basis. The answer to that really came in 1913 when Sturtevant, an undergraduate at Columbia working in the Morgan Fly Room, had one night, and I won't read this to you, but it talks about the idea he had and skipped his homework, because he was a sophomore in college at the time, to write up his idea of linkage mapping that said that the Morgan lab had figured out that there were multiple genes per chromosome and that they showed linkage. They weren't perfectly connected. There was recombination, so they would be disrupted, and that the distance on the chromosome was read out as the likelihood of a recombination between two markers. And what Sturtevant realized one night was that if you had a new gene that you'd found a mutation in, this is in fruit flies, and you studied its segregation, you could place it on a map based on the recombination patterns with markers of known position, and therefore identify exactly where in the genome the gene underlying that trait was without knowing anything about the gene whatsoever. And he wrote this up, and they understood the implications of this, that they could build genome maps, and if there were new mutations, they were thinking very much in that mindset, but really any genetic variation that has a map position can then be localized based on its recombination patterns, either in a family, in a single generation, or in a population based on ancestral recombination, which is what genome-wide association studies with common variants have done, and figure out what's going on. The thing about this idea was, although it was actually immediately put into practice in Drosophila, and to make maps of where the genes were, there wasn't for many years, I'm going to come back in a few minutes to what happened 60 years later, 6-0, that made it possible to actually identify the genes underlying these map positions, the DNA sequence variants. So this idea was there, and in fact was developed quite extensively in model systems. But the first case in which a human disease, a human genetic disease, was tied to a specific genotype, which was also the first time, by the way, a DNA change was linked to a protein change, was linked to a phenotype in any organism, was in the case of sickle cell anemia, where this man, Vern Ingram, following on work done by Linus Pauling, said, Linus Pauling had shown that there was a electrophoretic mobility difference of the hemoglobin of people with sickle cell than those without. And they had a paper in Science called Sickle Cell, a Molecular Disease, saying this was a disease of humans that was traceable to a difference in a protein. But actually the fact that they could show electrophoretic mobility didn't actually nail down where it was or that it was genetic. Vern Ingram went on to first show through biophysical methods just that this difference in the mobility of sickle cell, of beta globin and sickle cell anemia traced to a particular fragment of the protein, and then eventually sequenced the protein, not the DNA, but the protein, and found the sickle cell change. And this was one gene, one protein, one disease, and and Vern Ingram then spent his career at MIT just right across the street as a professor of biology. So again here, there is a deep insight into the nature of biology and into the basis of this disease, but also an approach that required here, this is a candidate gene study. Right, if you think about what the sickle cell story was, it was that red cells sickle. Red cells are bags of hemoglobin. So you purify the hemoglobin from red cells and run it on a gel. You see a difference. Then you study the one major protein. If it had turned out that sickle cell anemia was not due to beta globin, which was the majority protein, but some rare protein, or there wasn't a cell type that was easily purified, this discovery would not have been made, because you couldn't have done the candidate gene study of studying the hemoglobin without being able to purify it and know what you were studying. So coming back to the idea of linkage analysis from Morgan and fruit flies, that developed to the point where there were maps of many, many organisms, although as I'll describe soon, not of humans, but you couldn't get from a map position to a gene that was responsible until in 1972, David Hogness submitted a grant application that described in one grant application both the method of positional cloning that allowed you to merge the genetic map of where the variants, the inherited markers were, to the physical map of the genes and the DNA sequences. And this same grant application essentially described genomics and how the DNA sequence of the human genome would be determined in 30 years from this date. Because what he described in his grant was that you would take chromosomal DNA of the fruit fly, chromosome 4 in this case, and you'd break it into small clones that were subclones, pieces of this chromosome. And then you take each subclone and you'd break it into smaller subclones. So you'd make a map, and then you'd order these subclones. You'd make a map of pieces of DNA that you could say were in an ordered array that tiled across the chromosome. And then he said you would go ahead and you would sequence these. He said you do restriction mapping and you would do methods to identify sequence changes. And actually, 11 years later, Welcome Bender, who's a professor at Harvard Medical School, was the first author of the first paper that used this with David Hogness and Ed Lewis and others to identify specific genes for Drosophila homeotic transformations and specific mutations in those genes. And so this now put together with linkage mapping offered an approach to first get from the trait to map position, and then with positional cloning to get from map position to a gene and a sequence change. So this idea of genetic mapping and positional cloning in model systems really became then in the 1970s and 1980s, let's say, really and beyond, a mainstay of how you would dissect any biological phenomenon of interest. Is that you could take, in particular, if there was a inherited single gene trait, which if you're working in model systems, you could just do what everyone did, which was do mutagenesis, throw out the 99% of the individuals who didn't have a Mendelian phenotype, and then study those, and then in a biased but informative manner, only study those that gave a single gene clean phenotype. And then you could identify the genes doing this linkage mapping and positional cloning, and tremendous things were discovered. Multiple Nobel Prizes were awarded for discoveries made, although interestingly, no Nobel Prize for actually the underlying methodology of positional cloning that made it possible to do all this, which I think is wrong, actually. Should have been the case that without positional cloning and without, there would be none of these discoveries. And the other important point I want to make is that when saturation mutagenesis is performed, so you don't find one mutation and say that's the whole story in one gene, but you take a trait and you find as many mutations as you can until each gene is hit multiple times, then what you find in almost every case where people have followed through is coherence to the biology that emerges. It's not that you always get many genes, or almost always get many genes that can lead to the same trait, but it's not that they're like 25 different pathways with one mutation in each. It's almost always the case when you study them that there's clustering of mutations in given biological processes. And this is very important to our work in human genetics, because in many of our diseases, we're at a point in the discovery where we clearly have not saturated the pathways and the mutations, and we often have different variants, and we can't find the coherence yet. And it's possible there will never be any, but I don't believe that. I think it's simply that we haven't yet both reached a level of saturation where we've hit each pathway multiple enough times that we can recognize that there's a clustering, and second of all, that we don't actually know about most of the pathways, as was the case with this model systems work. That many of the things that you now would recognize as a well-understood canonical pathway, with all sorts of biological descriptions and narratives, started as a positional cloning study where people just had mutants with funny names, ultrabithorax, or SED3 or something. Names that didn't give you any biology. People found the genes, didn't know what they did. and then studied them for many years, and it was only that study for many years that revealed the biology, and now you can say very neatly and cleanly, you start with an organism, you get mutations, it's in this pathway, that leads to this disease or this phenotype, we understand it. That is the goal in some sense of what we're doing. So the trouble with doing this in the human population, I've been talking about model systems, is that first of all, having a genetic map required that there be markers that were inherited and could be traced through the generations of map positions. And so in Drosophila, these were things like curly wings or white eyes. They were phenotypic markers. In humans, they were phenotypic markers like ABO blood group or later the HLA markers. But there weren't generic markers at high enough density in humans so you could create a genetic map of the whole human genome. Also, obviously, as we know, humans tend to have small numbers of offspring and don't take lightly to being told who to mate with because it's an informative mating. And even if they did, it would take decades for you to do your experiment. So you're stuck with whatever matings exist in the population and a small number of offspring. Also, it's much easier to do genetic mapping if you start with an inbred, homozygous, isogenic line. So there's no genetic variation. Then you induce mutations at a rate such that each individual has 0 or 1 mutation, let's say. And then you take out just those individuals who have a clean phenotype, you throw the rest away, and then you just trace that one inherited mutation through the pedigrees. That's what people do in fruit flies, that's what people do in worms, that's what people do in mice, that's what people do in experimental models. Not surprisingly, the genetics of that turns out to be there's one gene in each cross that's segregating that affects the trait because you set it up that way. But humans are not isogenic. We each differ by millions of genetic variants from each other. And in each cross, there's a different set of backgrounds segregating. And so this is, and not surprisingly, given that you haven't set up the whole system and excluded everything that doesn't show complex inheritance, almost everything shows complex, not monogenic inheritance in terms of the average trait. Although there are, of course, many important Mendelian phenotypes that exist. So the thing that made it possible first to identify the genes underlying human single gene disorders was the recognition in 1980 or so that DNA polymorphisms themselves could be genetic markers. In other words, rather than using phenotypic markers, the phenotype could be a restriction and fragment length polymorphism on a gel, or later a length polymorphism, or later a SNP, or later a genome sequence. It doesn't matter. DNA sequence can itself be markers on a genetic map. And this led very rapidly to the first genetic map of human that was published in 1987, I believe it was. Remarkably, Mark Daly, who will be here shortly, who is my co-director of MPG here at the Broad and colleague at Mass General and Harvard. Mark is actually the second or third to last author on this paper, which is remarkable. Look how old he is. Subtract 1987 from 2012, which you'll see is 25 years ago, and we will return to how old Mark must have been when he was the second or third to last author on the first genetic map paper. It's something like 13 years old or something like that. No, he was like 20 or something like that. It's quite remarkable. Anyway, so this took about 15 years then from the first genetic map being published to its application through the paradigm I've described to you, leading to the discovery of thousands of genes and mutations underlying important medical conditions, and a complete rewriting of the textbook of Mendelian inheritance in man from a purely phenotypic description prior to this era, here's what the phenotype is, to a molecular and phenotypic description that could tell you here are the genes that are then mutated that can give rise, and here's how the different genotypes lead to different phenotypes, and as it evolved, here's the biology, and a remarkable transformation of a field. In the case of complex diseases like type 2 diabetes, there were Mendelian subtypes that had been recognized. So in other words, although most patients with diabetes have complex inheritance, about 1% of the patients are in families where there's early onset, single gene, autosomal dominant in this case, inheritance of diabetes, and those diseases were worked out with this approach and led to important insights as well, although the vast majority, in the case of diabetes, 99% of the inheritance of this disease was not explained by those studies. Moreover, extensive efforts to apply this approach of linkage mapping in large families and in isolated founder populations where the genetic complexity should be less really failed for complex disorders. So although this method I described has been spectacularly successful in the case of single gene disorders, it pretty much was a complete failure when it came to common diseases. And it's not that everyone predicted it would be a complete failure, because in the 1990s, this is much of what human geneticists did, was many human geneticists went out and did in diabetes and heart attack and schizophrenia and bipolar disease linkage studies. In well-designed studies, had there been single genes that had large effects on these traits, and they did not find them. So what next? So going back in time, remember I said how Darwin had put on the map the idea of phenotypic variation and its inheritance, and then how that would lead to natural selection. And it really bothered him that he didn't know how that worked. Where did that phenotypic variation come from, and how is it passed along? So his cousin, Galton, took up this challenge. And Galton was very aware he was Darwin's cousin. He said, this is the missing piece. He actually worked with Darwin on trying to figure out, how does this variation in traits work? And they did an experiment together where they parabiosed rabbits. And they took some rabbits that had different phenotypes, and then they stitched their bloodstream together, because Darwin had a theory about corpuscles in the blood being where this inherited matter was transferred, was carried along, and it didn't work. And so Galton scratched his head, and he had failed to show his famous cousin's idea was correct. And so what Galton ended up doing was literally going around saying, I'm going to start measuring things. And he measured heights. And he measured heights of parents and children. And he famously invented regression analysis. So he could put the heights of the parents and the heights of the children on the same plot. And he discovered that they were correlated. And the slope of the line of the relationship between the mid-parental height, the average height of the two parents, compared to the height of the children, is called, turned out to be the heritability, this concept of heritability. And so Galton, and they said, wow, they published this paper, and they said, well, so there's this interesting thing that they saw, because they weren't starting from a Mendelian or genetics view. They were just saying, here's what the trait variation is. And they found that although someone of a given tall parents could have tall or short children, and there was a big scatter, and short parents like me could have tall or short children, there was a scatter. In fact, my son, who just started high school today, love you, Zach, he's 5'10", and I'm 5'6", and I am his father, by the way, I can tell. And so short people like me can actually have tall kids. But on average, there's this very strong correlation, all right? So a movement developed called the biometric movement. The biometric movement did what it sounds like. They metriced bios. They went and measured living things, and they tried to understand these relationships of the traits of parents and traits of offspring, and the relationships of whether you were identical, this is the movement that sort of led to things like, as I started the talk with, identical twins and first-degree relatives and second-degree relatives. And they came up with a worldview that was kind of like paint colors, right? The idea that, like, if you imagine like tall and short is like how, different colors on a spectrum, and that the offspring were kind of like mixing the colors. So it was continuous variation, and you get a blending of characters, okay? This sounds nothing like Mendelian genetics, right? But it actually developed into a movement because it fit the data so well, okay? But then Mendel's rediscovered, right as the biometric movement is really doing well, and it leads to something called the biometric Mendelian debate, a vicious, ongoing debate that started literally in the first decade of the 20th century and rages on today in the American Society of Human Genetics in 2012, okay? Because one group of people said, I look at traits around me and they're not typically Mendelian inherited and there's blended characters. Another group of people says, well, but the individual genes and some traits are inherited according to Mendel's rules, and this is just one of those Rodney King, can't we all get along? Because the resolution to this debate was published in 1918. By R.A. Fisher, the great geneticist who invented statistics and the p-value. If you ever heard of the p-value, you can thank R.A. Fisher, okay? So what he wrote in 1918 in a paper called The Correlation Between Relatives, right? So that's the biometric part, under the supposition of Mendelian inheritance. That's saying Mendel's right. This is like Einstein. There was this famous thing about particles and waves. Is light a particle or light a wave? And Einstein famously said, well, light can be a particle and a wave. Okay? This is like, is it taste great or less filling? And R.A. Fisher said, well, it can taste great and be less filling. Okay? And the way that could work is if there are many different individual genetic contributors to a trait, each of which is inherited according to Mendelian pattern, but that they sum up in some quantitative matter to lead to any given trait. And if there's just one genotype that has a massive effect, that's what we call a Mendelian trait. And if there are many of them, or there's environmental other factors, it'll be a complex trait, and it's a single unified view. And if, as Popper said, the status of a scientific theory is attributable to how well, it escapes refutation by empirical observation in the years to come, R.A. Fisher's model's really a high status theory. Because now, almost 100 years later, I'm not aware of any observation in human genetics that doesn't actually fit this model essentially in its flexible form, but much more extreme views are refuted every day if you just pick up any journal, okay? So this model says that the genetics of complex traits, and therefore the common diseases that are typically complex, will involve many genetic variants, and they will act in a quantitative, not a deterministic manner. Those quantitative effects could be large, or they could be tiny, small, there could be many of them, there could be few of them, but that it will be quantitative and probabilistic rather than deterministic, all right, and unitary. It'll be polygenic and quantitative rather than single gene and deterministic. And the cases where it will be single gene and deterministic are probably recognizable a priori because they'll show that inheritance. And in a case where there is this probabilistic relationship between genotype and phenotype, you inevitably have to then compare frequencies in large groups. Because if the effect of phenotype and genotype is probabilistic, then it's not going to be sufficient to look at very small numbers of people and expect all or none. It's going to be something more fuzzy than that, right? And this is actually the part, and I said earlier, and there's a lot of people gathering, and so I may just sort of just cover a couple more things and then close out, because these are actually the important concepts. But this is the part where I think, remember I said early on, it's like the political conventions where there's a reality, but different people describe it in different ways. This is, I think, maybe where the deep issue resides in our field, is that to some people, genetics means there are traits and they vary and I want to understand that, or there's biology and I want to understand it and genetic mapping is just a tool to find the genes. And to some people it's, as I said, it's single gene disorders and they want to understand that. But at some level, the word genetics means to some people simple and deterministic. And so when things are observed that are not simple and deterministic, it seems like genetics didn't work or it failed or it's unsatisfactory. But the message I would have is genetics is not actually, in most cases, simple and deterministic. It's complex, but it's incredibly powerful. And so what you're going to hear about in the months to come, in addition to cases of single gene disorders that are incredibly powerful and important and clinically moving forward, is many cases where first using common genetic variants and now genome sequencing that has every genetic variant, people are trying to understand this relationship of genotype and phenotype. And then that'll be part of what we talk about. And one of the things that's happening in our field today is that the tools of DNA sequencing have gotten so powerful that now we can really collect all the data in a genome in large samples. So we not have to work with slices of the data. We can have all the data. And we can try to understand all the genetic variants that can be observed and their contribution to disease, common and rare. And that'll be maybe, half-ish of what we talk about is people trying to do that in different diseases and getting different answers and what have you. And in addition, we're going to talk a lot in this series about how do you get from those observations to biological understanding. Because if, as I said earlier, if all we do is get this mapping of genotype and phenotype, we'll have learned a lot. Just as simply knowing which traits have different genetic properties and what their phenotypes are taught us a lot. But if we can't get from identifying the genes in this unbiased way, which is the sine qua non of our field, if we can't get from discovering them in an unbiased way, this new knowledge, to new understanding, and then ultimately new application, we won't have done anything. That's going to take a long time, but we're on the road today. So with that, I'm going to stop. Chris, I don't know if you want to say anything before we move on.
Great. Thanks very much, David. If there are any questions, I know David would love to answer them. Down in the front, we're going to stop and let folks file in. And as a reminder, next week there will not be a primer. 2 weeks from today, Jim Gasello will be talking about Mendelian genetics. But again, you'll realize that in David's Zen analogy, this is less like the Democratic and Republican realities and more like a convergence on the same reality of overall human genetics.
Absolutely.
Thanks very much.
Thanks.